In this PowerPoint, we are going to be focusing on gene mutations. And from page 20 of your student guide, we're going to be looking at the first 4 learning intentions. So you need to be able to explain what a mutation is, identify what is meant by the term single gene mutations and how they are brought about, explain what is meant by the terms missense, nonsense, and splice site with relation to nucleotide substitution mutations, and finally explain the consequence of nucleotide insertion and deletions with regards to being frame shift mutations. We have covered mutations in a bit of detail at National 5 level, so just as a reminder, a mutation is defined as a random change to genetic material. Mutations may be neutral, that means they have no noticeable effect on the organism, or they could give rise to an advantage or a disadvantage. It's important to know that mutations are spontaneous and are the only source of new alleles or new variation within a species. Environmental factors such as radiation and some chemicals can increase the rate of mutation. A mutation is a change in the structure or composition of an organism's genome, and a genome is just characterised by the genetic information that each organism has. Mutations vary in form from being a tiny change in DNA structure of a gene to a large-scale alteration in chromosome structure or number. When such a change in genotype produces a physical change, like something you can see in the phenotype, the individual affected is referred to as being a mutant. As we had a look at in National 5, but just to revisit, gene mutations arise spontaneously and at random, and they occur rarely. When exposed to mutagenic agents, yes, they do increase in frequency, but aside from having mutagenic agents present, mutations are actually quite a rare and random occurrence. A condition or disease that can be shown to be directly related to an individual's genetic information is called a genetic disorder. For a protein to function properly, it must possess the correct sequence of amino acids determined by the order of the nucleotide bases on a particular region of DNA in a gene. So we've been looking at that in the last couple of chapters, having a look at the impact of the DNA sequence and the effect that it has on the amino acid sequence. So what we're looking at when we're talking about a genetic disorder is that the sequence of bases would be altered, so therefore the amino acid sequence would be altered in the protein. A change to the gene or a chromosome caused by a mutation may result in the gene expressing a faulty version of the protein that does not function properly. The mutated gene may even fail to express a protein at all. While you're on this slide, if you click on the image, it should take you to a video link which gives you an overview of all the types of mutations we're going to cover in this chapter. We're going to concentrate in this PowerPoint looking at just single gene mutations. Now, single gene mutations involve an alteration of a nucleotide sequence in the gene's DNA. We refer to them as being a point mutation, as these are a type of single gene mutation that involves a change in one nucleotide in the DNA sequence of the gene. There are 3 types of point mutation that we cover at higher human biology, and they're either called substitution, insertion, or deletion. In each case, the point mutation results in one or more codons for one or more amino acids being altered. The first of the single gene mutations we are going to focus on is called a substitution mutation. Now, when we look at a substitution mutation, what we see is that one nucleotide is replaced or substituted with another nucleotide. The effect of the substitution mutation is that it will affect one codon only, so we regard this as being a point mutation. The impact on the protein structure, however, can be divided into three categories. We can see the impact of this single point mutation as being either a missense effect on the protein, a nonsense effect on the protein, or what we call a splice site mutation. Taking each one of these in turn, and remember each one of these categories is caused by a substitution of one single nucleotide for another. So following a substitution mutation, the altered codon codes for an amino acid that still makes sense but is not the original one. This change in the genome is called a missense mutation. So for example, if the substitution of a guanine for a cytosine base still codes for a particular amino acid, it's not the correct one, we would call that a missense mutation. Moving on to nonsense, as a result again of a substitution mutation, a codon that used to code for an amino acid is exchanged for one that acts as a premature stop codon. It causes protein synthesis to be halted prematurely, and this results in the formation of a polypeptide chain that is shorter than the normal one and is unable to function. Hence why we call it a nonsense mutation, because although we've got a bit of a protein chain, it's a lot shorter than it should be. So technically, it makes up a nonsense protein. Finally, having a look at the impact of a single substitution mutation giving rise to what we call a splice site mutation. So if one or more introns have been retained by the mature transcript of the messenger RNA, they may in turn be translated into an altered protein that does not function properly. So if the splice sites are affected and introns start to be retained, we will see a faulty protein as a consequence of this substitution mutation. When one of the genes on chromosome 11 that codes for hemoglobin undergoes a substitution, it becomes expressed as an unusual form of hemoglobin called hemoglobin S. This is an example of a missense. Although hemoglobin S differs from normal hemoglobin by only one single amino acid, that tiny alteration leads to profound changes in the folding of the protein and ultimately the shape of the hemoglobin molecule. Hemoglobin S is inefficient at carrying oxygen, which is obviously the prime function of hemoglobin within a healthy red blood cell. So what we see with sickle cell disease is that the red blood cells are less flexible than the normal type or the healthy red blood cells, and they tend to stick together and can interfere with blood circulation. This can result in problems regards things like your oxygen, severe oxygen shortage, going into vital organs, and if that's significantly affected, then you might see damage, long-term consequences to these vital organs, and can result in death if it goes by, obviously, undetected and untreated. Phenylalanine and tyrosine are two amino acids that human beings obtain from protein in their diet. During our normal metabolism, excess phenylalanine is acted on by an enzyme which breaks it down into other substances that the body can use to manufacture tyrosine. Phenylketonuria, or PKU as it's abbreviated to, is a genetic disorder caused by a mutation to a gene on chromosome 12 that normally codes for enzyme 1 in the metabolic pathway that sees the breakdown of phenylalanine to tyrosine. Most commonly, the mutated gene has undergone a substitution of a nucleotide and missense occurs. The altered form of the protein expressed contains a copy of tryptophan in place of arginine, and it therefore becomes non-functional. So the final enzyme that's trying to be manufactured as a result of the substitution mutation is going to bring about a non-functional protein. As a result of this single base substitution or single gene substitution mutation, the individual gets what's called an inborn error of metabolism, which is kind of shown by the diagram in front of you, which means that phenylalanine is no longer converted to tyrosine, and instead it accumulates and some of it is converted to toxins. These toxins inhibit some enzymes that are involved in chemical reactions and biochemical pathways within brain cells. So therefore it can affect brain development in early years and can result in individuals who have PKU undetected as having severe learning difficulties. In Britain, PKU is screened for at birth, and if it's detected, the individuals are put on a diet containing minimum phenylalanine, and that reduces the impact of the condition. Duchenne muscular dystrophy is caused by any one of several types of mutation to a particular gene on the X chromosome, such as a deletion or a nonsense mutation. The affected gene fails to code for a protein called dystrophin, which is essential for the normal functioning of muscles. In skeletal and cardiac muscle, for example, dystrophin is part of a group of proteins that strengthen the muscle fibers and protect them from injury during contraction and relaxation. Duchenne muscular dystrophy is the most common form of muscular dystrophy, and in the absence of the dystrophin, skeletal muscles become weak and lose their normal structure. What you see with this condition is a progressive loss of coordination. People with this condition are severely disabled from an early age and normally die young without passing the mutant allele on to the next generation. Duchenne muscular dystrophy is known as a sex-linked condition, which we'll look at in more detail in Unit 2, and is almost entirely restricted to males, being passed on by carrier mothers to their sons. Beta thalassemia is a genetic disorder caused by several different types of mutation that affect a gene on chromosome 11 that codes for what's called beta globin. One of the most common of these mutations is a substitution mutation that we've been looking at that occurs at a splice site on an intron, and it causes the base guanine to be replaced by a base adenine. One of the examples of beta thalassemia is characterized by the complete lack of production of beta globin. And what we see in place of it is the production of a totally altered version of the protein. The person in this instance has excess of certain proteins in their bloodstream, which tends to bind to and damage red blood cells. Patients with severe beta thalassemia require medical treatment such as blood transfusions. We're going to look now at the two remaining gene mutations, which are called insertion and deletion. Hopefully their names would be self-explanatory in the fact that insertion involves the addition of a nucleotide into the base sequence, and deletion sees the removal of a nucleotide from the DNA base sequence. Because mRNA is read as a series of triplet codons during translation, if one base pair is inserted or deleted, this affects the reading frame or that triplet grouping of the genetic code. Whether it's insertion or deletion, that triplet grouping is going to become shifted in a way that alters every subsequent codon and amino acid coded all along the remaining length of the gene. The protein formed as a result of these frame shift mutations is almost certain to be non-functional. Frame shift mutations cause all of the codons after the point of mutation to be altered. So they either all move up one if it's been an insertion mutation, or they all move down one if it's been a deletion mutation. So if all of the codons have been affected, that therefore means when we come to translation, all of the amino acids will also be affected. Tay-Sachs disease is a genetic disorder resulting from a mutation to a gene on chromosome 15. Under normal circumstances, the gene is responsible for encoding an enzyme that controls an essential biochemical reaction in nerve cells. Changes to the gene take the form of point mutations, such as an insertion or deletion, which result in, as we know, a frame shift effect. The protein expressed is so different from the normal one that it is non-functional. As a result, the enzyme's unprocessed substrate accumulates in brain cells. This leads to neurological degeneration, generalized paralysis, and death at about 4 years of age. Cystic fibrosis is a genetic disorder caused by a 3 base pair deletion to a gene on chromosome 7. This type of mutation removes a codon for phenylalanine and causes the coded message to be seriously altered by the frame shift effect and produce a non-functional protein. The normal allele for the gene codes for a membrane protein that assists in the transport of chloride ions into and out of cells. In the absence of this protein, an abnormally high concentration of chloride gathers outside cells. Those regions of the body that coat their cells with mucus become affected because the high concentration of chloride causes mucus to become thicker and stickier. So organs such as the lungs, the pancreas, and the alimentary canal become congested and blocked. Individuals have to undergo regular pounding on the chest to clear the thick mucus, and daily use of antibiotics can extend the person's life into their 30s and beyond. Untreated, an individual with cystic fibrosis would normally die at the age between 4 and 5 years. I would ask you to evaluate your own learning based on the 4 learning intentions that we've covered during this PowerPoint as shown on the screen.